Welcome, class. Welcome to Size Chromatography
Hello,
101.
hello,
Today we're going to be
Professor.
learning about, you guessed it, size chromatography.
Yeah.
Yeah.
Size exclusion chromatography.
Oh,
So,
importante.
the purpose for size exclusion chromatography is to be able to separate different molecules and proteins based off of their size. For instance, if you look on the right, we have our red proteins. The small protein at the bottom here, because it's so small, it's going to get stuck inside the special gel that is
Mm,
in our column.
mm.
And the gel is really porous, like the sponge over here. So our small protein is going to get stuck in the pores, and it's going to take a while for it to move down to the bottom. But our larger proteins, like the middle one and the top one, they're not going to get stuck in these pores as easily as our small one. So they're
Ah,
going to be able to go through the column much faster.
got it.
Yes. And as you can see, this is our column apparatus. It is filled with gel and being held up by a ring stand. So let's move on to our pre-lab demonstration.
Who are these
So
fine fellows?
these are how we're going to represent our molecules and proteins
Hmm.
based off of size. As you can see here, our baby zombie is very small. He is just a little bit over 1 block tall.
Oh, he's
And then
like
we
the
have,
shoot over there.
he is.
Ah.
And so our next item of interest is Mr. Creeper.
Mm.
He is he is only a block and a half tall. And then we have
Mm.
Nice.
the man, the myth, the legend, the zombie, classic. Um, he's about 2 blocks tall.
I see.
And then we have our friend, the wither skeleton, who is 3 blocks tall. So all of our we're just gonna call them proteins. All of our proteins
Mr.
are at
Protein.
different sizes. And so we're going to be able to um, visualize them going out through their column.
Got it.
So, any questions before we go on?
Not a single thought.
No. I'm learning.
Perfect.
Yay,
Alrighty,
experiment.
class, let's go on our field trip.
Experiment time, experiment time.
Yay.
Whoa, they're up
Why he
there.
sit like
Oh,
that?
He's my
hey,
boy.
no
Whoa.
animals,
Whoa.
Oh, I'm gonna turn.
no animals in the
Oh.
classroom.
We're gonna turn away from that. We can just do a snap cut the
hmm
outside.
Alrighty, class. So I have set up our makeshift size exclusion column.
Ooh.
And as you can see up here, we have our gang of proteins,
Oh, protein gang.
and the um, this water stream is going to simulate gravity, um, pulling the pulling the proteins from the top of the column down to the end. And where all of our orange blocks are, these are going to be um, representing the pores of
Oh,
which proteins get stuck in.
like the sponge.
Right. So
What?
does anyone have any thoughts on why we need to separate our proteins by size. Any thoughts?
To isolate a
Mhm.
certain protein.
Correct.
Wow.
Correct. Here, you get a bread.
Wow, smarty
Oh,
pants.
haven't eaten today.
Yeah, so in lots of pharmaceutical or even analytical um, experiments, we would need to isolate a specific protein uh, from a source that it may be from cow liver or from the venom of a snake, and we would need to isolate a specific one. And since we would know what weight it is or how big it is,
Mm.
and then we would be able to send our molecules or our proteins through our gel, and we'd be able to pick out the
I want the
our
creeper.
proper yeah right, our proper protein that we want.
I want the creeper.
So let's begin our little demonstration. So this is to simulate adding just a whole solution of random proteins. So
Mm.
let's see. So we look at first our smaller molecules are being held up in the pores,
Oh.
and our slightly bigger ones here, like the creeper.
You got
And now the
stuck.
zombies can fit into this pore, but the large wither skeleton cannot.
Here. Like the
hmm
sponge.
Goes all the way to the end. And there we go. We have successfully isolated all of our proteins. Now,
Yeah.
now this demonstration is just to show the separation of the proteins. So this is like if we were running our column, um, and we were take like a snapshot in time, our column would look something like this. But if we kept our column running, our wither skeleton, our large protein, would come out first, and then
Yeah.
all of our other proteins would slowly move out the column, and then our next one would be our zombie protein coming out, then our creeper
Mm.
protein, and then last our baby zombie protein.
I see.
And
Yeah.
that is the gist of size exclusion chromatography. I think that
Woohoo!
so thank you all for this great lesson. You guys were wonderful students, except for the one with ADHD over there. And that's gonna
And
wrap up our
that's gonna,
class.
yeah,
Thank you
that would be
all.
class.
Yeah.
Yay.